Make way, coming through. In this lesson, we're going to talk about the parkouriest of the white blood cells, the neutrophils. And because neutrophils are known as the first responders of the immune system, we've symbolized them with our recurring first responders. You'll see this symbol not just throughout immunology, but throughout all of Sketchy. Now, neutrophils, which you'll sometimes hear referred to as PMNs, are the most abundant of all circulating white blood cells. They make up about 70% of the total leukocyte population. And this responder is running into our scene because these cells respond very rapidly. They're the most abundant during the first 1 to 2 days after an infection or injury. On the flip side, though, they have a short lifespan relative to other immune cells. And unlike macrophages, neutrophils are not antigen-presenting cells. As you'll soon see, their main job is to eat, aka phagocytose, bad guys. At rest, neutrophils circulate in the blood, which is why this running responder is in one of our castle's many ominous subterranean red passageways. But as you can see from our sketch, a first responder in one of these tunnels is nowhere near the potential invaders that come in from the outside. And so we're going to spend most of this sketch actually talking about how neutrophils get from the blood to the site of infection or injury. First, the neutrophil needs to know where the infection is. But last time I looked, they don't have eyeballs. Doesn't matter. A well-coordinated system of chemokines and sticky molecules on the surface of capillary endothelial cells and on the neutrophil itself help it get where it needs to go. So let's talk through this process a little bit. First, a macrophage, and if you remember, they're the sentinels of the body, recognizes an invader. So we've got our macrocage kid here, and he's spotted our recurring invaders. See that they've got those pamplemousses off in the distance? Once the macrophage recognizes an invader, it secretes cytokines, things like TNF and IL-1, in order to alert other immune cells, in this case neutrophils, to come to the invasion. TNF is symbolized by a recurring tuna fish, while we've made IL-1 unforgettable with this IL-1 iguana. Remember that all of our interleukins are a bright neon yellow to symbolize their signaling function. Then, in response to these cytokines, vascular endothelial cells near the site of infection begin to express selectin. You might also see that just abbreviated as SEL, like what we've written on this button in the endothelium of the tunnel. Selectins are actually a class of adhesion molecules, and the specific selectins expressed by endothelial cells at this point in the migration process are technically P-selectin and E-selectin. But I'd way rather you remember just selectin in general than one of these specific ones, especially because we've got, unfortunately, some more alphabet soup to come. Okay, here comes the fun part. You see that awesome parkour move? Let's go through that in a little bit of slow motion. So I spent all that time talking about selectin because neutrophils have a molecule on their surface known as a selectin ligand, or a SLIG. Sounds kind of disgusting, but we'll represent it with this sling that our neutrophil EMT is wielding. When selectin and the SLIG get near each other, they want to bind, and that causes the neutrophil to slow down and start rolling. See how our first responder is using their sling to hang onto the SEL button to pull off that gravity-defying move? It's important to note here that this binding between selectin and SLIG isn't too tight. It can also be called a low affinity adhesion, or what I prefer to call it, the slow your roll, dude. Because they've now slowed down, these neutrophils come into more contact with chemokines from the injured and inflamed tissue, such as C5a and lipopolysaccharide. Neutrophils take those molecules as signals to push more integrin to their cell surface. And there's our integrin, an integral-shaped hook wielded by our first responder. I know you weren't expecting to think about calculus today, but the integral just has that classic shape, and it's a perfect fit for this symbol. Now, I know integrins feels like just another molecule to memorize, but they're actually kind of important. And in fact, we're going to double down. We're going to name one of these integrins called CD18. A deficiency in CD18 leads to leukocyte adhesion deficiency syndrome, and so it's kind of important to remember that. So we'll draw in a little sign reminding you that the contents of these barrels are 18 and over to help you keep that number in your mind. So a castle's security tunnel system isn't complete without a security camera. And even though that camera doesn't look very useful because a robber would have to hold still for like 5 hours for them to get a picture, that camera is there as a symbol for ICAM. ICAM is expressed on the surface of endothelial cells, and it's the ligand for integrins. So when integrins and ICAMs come together, well, then we've got a real sticky situation on our hands. The bond between integrin and ICAM is very, very strong, and in fact, it's strong enough that it causes the neutrophil to come to a complete stop. And a stopped PMN is exactly what we need so that the neutrophil can change direction and exit the tissue toward its target, which is the site of infection. It does this via binding to PCAM1, that stands for platelet endothelial cell adhesion molecule, which is found within intracellular junctions on the surface of endothelial cells. And finally, we've got an exit out of this frickin' tunnel. A pecan tree root, to be specific, and that should help you remember PCAM1. Because PCAM1 is located in intracellular junctions, it's in a place where the PMNs can kind of squeeze through those cells and out of the blood vessels, or in this case, out of the tunnel, and get another step closer to the enemy. And now the neutrophil is unleashed into the tissues. It's generally in the right spot, as in it made it to the right city out of all the cities that exist, but we still need to get an exact address. As you may have guessed, we need more chemoattractants. The same types of molecules that cause the neutrophil to display and activate their integrins also double as signals to where the infection is, and as a reminder, that was C5a and lipopolysaccharides. Similarly, macrophages that have already encountered the pathogen will have released IL-8, which we'll symbolize with this 8-shaped owl. That's another signal that triggers neutrophils to migrate to the site of infection, and it's also a signal for them to start chowing down, aka phagocytosing. And finally, that brings us to the function of neutrophils. They actually have a ton, but as we mentioned at the beginning, primary among them is to kill enemy cells via phagocytosis. Chomp, chomp, chomp. Yummy. But ever the team player, neutrophils also secrete cytokines, represented by this signal flare, which help recruit more cells to the site of infection. An example of one of these cytokines is our old friend, TNF. Also, if you remember way back to our cells of the immune system lesson, neutrophils are granulocytes. That means that just like this first responder can throw grenades, neutrophils can secrete granules that are full of enzymes that can break down invaders, blow them up. And finally, neutrophils also have an extra special power that other cells don't have. Just like Spider-Man, they can shoot nets, that is, neutrophil extracellular traps, which has got to be an all-time acronym. Dying neutrophils release their nuclear contents, which kills extracellular microbes. Hooray! Because neutrophils are the first responders, they swarm to the site of infection in abundance. They don't live very long, but man, do they lead meaningful lives. Now, you may ask, what happens to all those neutrophils as they start to die? Well, have you ever seen a wound with white pus coming out of it? It may look gross, but that pus is made up mostly of neutrophils and a protein-rich fluid. So that may sound gross, but what's even grosser is a bunch of old, disgusting white coats. So we'll leave this pile of discarded white coats over here on the hill as a parting gift. And since we love ending on images that'll totally ruin your lunch, we're going to stop there. But before we go, let's do one quick review. Neutrophils are the first responders of the immune system and are the most abundant of all the circulating leukocytes. They can even be seen by the naked eye when they accumulate in the substance we know as pus. At rest, they circulate in the blood and need to migrate out of blood vessels in order to get to the site of infection to kill bad guys. Neutrophils do this via a multi-step migration process that is triggered by chemokines. When a pathogen is encountered, IL-1 and TNF-alpha are released by macrophages, which causes endothelial cells to express selectin. Neutrophils have a molecule on their surface called selectin ligand, or SLIG, which binds with low affinity to selectin on the endothelial cells, effectively slowing the neutrophil down. During this slow rolling period, the neutrophil also detects more signs of infection and injury, such as C5a or lipopolysaccharide, and expresses integrin on its surface. Integrin on the neutrophil binds to ICAM on endothelial cells with a very high affinity. This stops the neutrophil's movement completely. And remember that a specific integrin in this process is CD18, which is what is deficient in leukocyte adhesion deficiency. Once stopped, the neutrophil squeezes out of the vasculature by adhering to PCAM1, a molecule found in intracellular junctions on the surface of endothelial cells. And finally, more chemokines, such as IL-8, draw the neutrophil to its final battle site, and also trigger them to go into hyperphagocytosis mode. In addition to phagocytosis, neutrophils also secrete granules and nets to further aid in killing invaders. Also, they secrete cytokines such as TNF-alpha to recruit other immune cells. And that's it, we made it. Now tell me how a neutrophil migrates out of the blood vessels using only letters and numbers.